Good afternoon, everyone, and welcome to this month's SCI talk entitled AI in Drug Discovery with Professor John Overington. I am Paul Hernandez, and I'll be chairing today's event. This is the last of a series of events focused on the impact of AI in chemistry using industries. And it began in February with Professor Jeremy Frey's introduction. It was followed by Professor Ruth Meisner last month on Bayesian optimization and design of experiments. And today we'll be hearing about the many ways in which artificial intelligence can impact drug discovery. For instance, through its application in structural modeling, target discovery, or lead generation. Before that, a quick note on housekeeping. There will be a Q&A session at the end of the talk, but just make sure you submit your questions clicking the Q&A tab below. And all attendees are in listen-only mode, so I'll be answering the questions. So before we start, some quick introductions. I am Paul Hernandez. I am a postdoctoral researcher at the University of Oxford, doing medicinal chemistry and a lecturer. My work is focused on the development of new metabolic modulators with Novo Nordisk, and I am part of the organizing committee of SCITalk. It is a pleasure to introduce today's speaker, Professor John Overington, Scientist Chief Data Officer. John has over 30 years of experience in drug discovery across various sectors, including large pharma, biotech, and academia. Following his PhD at Birkbeck College, University of London, and postdoctoral stint at Imperial Cancer Research Fund, John joined Pfizer. This was followed by leadership positions at Informatica, EMBL ABI, Benevolent AI, and just prior to joining Cynthia at Medicine's Discovery Catapult. John has a track record of innovation and collaborative science and has developed several of the foundation data resources for AI-based drug discovery. So without further ado, I'll hand over to John for this lecture on AI drug discovery.
Thank you so much, and also for the flattering introduction that I wrote myself. I'm speaking to you from Houston, so I work remotely for Exscientia. I sort of lead the platform design elements of the organization. And today, given the sort of the previous speakers, decided to focus more on the sort of approaches that we use to apply AI techniques in our business setting. And for me, one of the most fantastic sort of features and enjoyable things of working in the drug discovery sector is that it really is I think one of the key touch points between science innovation, and industrial and medical benefit. So this science and industry link is I think, yeah, for me, an incredibly exciting thing. So happy to take questions at the end or anything a little bit afterwards. And hopefully now the first slide. So Exscientia is a company really built around patient-first AI. We've developed and continue to develop a series of integrated technologies, so data and algorithms and scalable workflows and software engineering to discover and design precision medicines. So what do we mean by a precision medicine? So this is something that we can increase. This is a program or a drug in which we can increase the probability of success by understanding the patient's system. And probably, for me, one of the great pinnacles of science of the last century and most recent sort of 20 years has been the Human Genome Project, and in particular the Cancer Genome Project, where now we can routinely genotype, understand the genetic mutations, understand even the sort of drug resistance, acquired adaptations, and often mutations that are part of the continuing sort of warfare of drugs and tumors. And there's a lot of similarities between some of the technical challenges in multiple drug resistance or acquired drug resistance in antibacterials and anti-cancers. There's many similarities of the underlying sort of systems biology. Also at the center of the company is this concept of encode and automating, capturing data in a computable form, developing hypotheses on top of that using AI and data science approaches. And then, as you'll see a little bit later, really coupling that design to a very tight integration with automation platforms. Drug discovery, I'm sure you know the sort of general numbers, but every time there's an analysis, they go up. But the rough cost of each new drug approved is about $2.3 billion per drug. And in many other areas of business and industry, automation has transformed the cost base. We certainly believe that engineering and automating drug discovery will make a huge productivity and also cost effectiveness. We have an internal pipeline, so we focus in oncology, ourselves for precision oncology. CDK7, LSD1, and MALT1 are the sort of clinic-ready or in clinical trials targets that we've had and developed solely in-house using our own pipeline, and then a series of partnerships. An example of which would be, say, the compound currently in clinical development, PKC theta, with Bristol Myers Squibb. So we've found, discovered, invented, designed 8 precision medicine drug development candidates. We've had 6 projects in licensed by partners. So, you know, the designs are quality and they do things in predictive models. We're behind the first clinical trial showing improvements in cancer treatment through the application of AI. We've got consistently a 70% reduction in discovery time from target ID to candidate, about an 80% improved capital efficiency in overall drug discovery sort of costs, and then 4 billion potential, you know, to be clear, potential in milestones negotiated with partners. So part of the success of the company, we were some of the very first entrants into using sort of generative methods for drug discovery. There was a foundation paper from Andrew Hopkins, Jeremy Besnard, in using generative methods to explore and then optimize chemical space. And we've been building on top of that initial academic science since in the successful implementation of our business. There's 4 ways that we can improve the probability of success. And as a little bit of background to this, as I'm sure many of you know, a lot of that 2.3 billion cost per drug is on funding failures. So many drug targets don't deliver attractive drug-like leads. You know, there's this sort of concept of druggability or tractability. Often the targets aren't linked to modulating the disease process, and because of the way that things are currently configured with randomized clinical trials, we often find that as an industry in a phase 2 efficacy study with a lot of sunk costs and, you know, none or only very minor modulation of the disease biomarker or pathology endpoint. So we are good. Some of the core strengths are in drug design. So developing compounds that address known issues with the compounds of others or the current state-of-the-art treatment approaches. To do this, we use a multi-parameter optimization and MPO approach to really sort of measure the improvement in design quality and efficacy at every cycle. Target biology. We have 2 approaches here. The first is an in silico AI approach to come up with better predictive associations of a target with disease pathology. And second, you know, to have very close to human preclinical models to ensure the accuracy or the likely probability of success in the clinic. Patient selection, especially important in oncology settings where different underlying causes can lead to the same basic overall presentation of lung cancer, bowel cancer, and so forth. So clearly understanding the genetic basis of this, or the pathway basis of this, and then therapies that target particular subsets is becoming du jour in the sector. And also clinical trials are often very expensive. Recruitment is often an issue, and we've developed some approaches that unfortunately I won't talk about today, where we're using AI in smart design to really minimize the cost and maximize the speed of clinical readout on the projects we work on. So a major part of what we do is just integrating knowledge of the molecular systems, chemical space, patent space, all sorts of aspects into a holistic view. We generate proprietary data ourselves from patient samples. We do a lot of work in target identification. Then some of the core technologies around AI, this generative AI design approach, model-based learning, active learning on the basis of the experimental results we get in preclinical models, the association of these with patient-derived biomarkers, and then this intelligent clinical design. And together, this is our sort of view, our AI-driven, AI-enabled view of drug discovery using our platform. Why are our clinical candidates different? So CDK7, we've got a precision design there. That the differentiation was primarily around the sort of tuning of the PK/PD, really specific for the target mechanism and function. PKC theta, again through design, precision design, and understanding, again sort of ADMET or PK parameters, getting a low predicted dose and hopefully optimal therapeutic index. LSD1, again, some of these broad acting or broad potential acting cancer targets, a big issue in some of the non-potency aspects of drug design, and again in MALT1, again addressing toxicity liabilities with other competitor compounds and so forth. And I think a key point to take away here is that a lot of the applications of AI in drug discovery are focusing on the sort of bimolecular drug target interactions, things like potency. But for a successful drug, it's often the PK, the exposure, the tissue distribution, the metabolism, and so forth that actually differentiates a quality drug and a quality drug candidate from a potent inhibitor. And so again, there's a general theme with how our use of AI contrasts with that of some others. The core thing to introduce here is this progression from target identification, verification, validation, through to this closed loop, this small molecule design and automation cycle, where the majority, I guess, of the slides will focus, give you details on what we do there, and then feeding into patient tissue selection and screening, and then feeding back into potential new target discovery opportunities. So that's the encode and automate cycle, with a big focus in this presentation on the design and the automation aspects. So target selection, what do? We have built an AI system of knowledge graphs and then transitioning these to LLMs. So we do a lot of text mining, the literature, the public literature, and also proprietary data sources that we've assembled, and couple this with a sort of strong ontology and organizational framework for the identity of the object, so we can resolve compounds or compound names back to chemical structures very simply, and then do computations on, instead of similarity of the gene name, we can do similarity searches at the compound level and within those systems. And like many in the field, we've been, I was stunned by the progress and efficacy of these large language models, these LLMs. But fundamentally, in our view, you need to couple knowledge graph, semantic web technologies with LLMs to have sort of effectiveness. A couple of other points to highlight here is that we've got several experimental teams to perform experimental target validation on the back of the some of the AI methods proposing or operating as a hypothesis generation technique for the targets that we work on. I see sometimes in the literature this being a little bit confused with this sort of hallucination process or hallucination viaduct or side effect. Viaduct clearly was a hallucination there in the neural network in my brain. So this hallucination issue, inventing or filling in, or supplying reasonable examples or reasonable hypotheses that just are fictitious, is a liability. Again, we've developed some recent technology to minimize that I think I'll show you in a second. So here's an example of how prompt tuning can increase or change the output of some of our methods. Where you add additional context, so domain-specific training and domain-specific knowledge and sort of target functions or target requirements in the query, you can really modulate the ranking or the likelihood of these targets being successful. And in our broader AI platform, all of these targets, every gene in the human genome, every gene in say malaria and viruses of interest to us as well, has got pre-computed tractability assessments. So what's the likelihood of this being a good target based on what we know about the properties of historically successful drug targets and drugs. So this is an example of where we sort of use a tuned LLM for target hypothesis generation. The model then looks, it's quite a smart system. You have a large sort of hypothesis space set. The model then generates queries to verify those statements. And then through that process, we find a lot of filtering to more consistent and stronger messages from the underlying data. So a broad range approach, an adaptive approach, an active learning approach to use LLMs for hypothesis generation, then use the same technology with a different slant to verify or check, to seek further verification and sort of provenance of those statements or hypotheses, and then a consistent answer that helps substantially in target identification. Now moving on to the design aspects, which we call precision design. Fundamentally, drug discovery is a learning problem. You follow a curriculum, a series of discrete steps where you have some prior knowledge, you integrate that, you come up with a drug design, a specific design, you test that, and then you see whether you're moving forward towards your eventual goal. It's just simply impossible to literally take a target, press a button, and then develop a sort of qualified clinical candidate with high probability of success. But I must admit, I do have dreams about that still, and maybe one day it will happen. So when you're operating this sort of iterative, sort of 20 questions sort of like game of moving towards a drug candidate, some of the important things to consider are how fast you're learning. We want to learn in the most efficient way possible, and also how do we quantify the information content in this optimization towards the target function, which is the properties of this target product profile, the pharmaceutical product-like characteristics of the small molecule, of which potency is often a very small or relatively minor component. It's necessary, but certainly not not sufficient. And we found that, and others have found, of course, that active learning is a very powerful technique to couple with AI approaches. And we've got some really nice examples now where we've used active learning to sort of break out of established areas of chemical space and move into areas of novelty, and also trying to quantify against the background of the inherent uncertainty in the biology and the chemistry results, how much each compound is adding in our sort of forward steps. Oh, sorry for that. I've mentioned data generation in the preamble. We've got extensive in-house proprietary generation data capabilities. So primary tissue disease models, data on certain molecular systems incredibly quickly. We also use high throughput crystallography. The more you know about your target, the more you know about the binding pose and so forth, the better. And again, like many others, our work has been really enabled by some of the breakthroughs in AlphaFold and this sort of these novel co-folding AI algorithm approaches, which again are sort of fabulous. And then also we've got, as part of our sort of automation suite that I'll come on to later, some AI embedded in the automated assay development, the very rapid establishment of the screening assays, the validation assays, positive, negative controls, and so forth. So this is the internal proprietary data generating capability, but this is clearly built on a lot of the public domain data. One of the earlier sort of projects in my career that I was very happy to lead is the Kemble data resource, which arguably transformed the ability for the AI drug discovery sector to be sort of birthed. It gave the community examples and training and test data that clearly have led to a lot of innovation around target prediction, generative and genetic algorithm optimization of compounds, and so forth. So I think proprietary data and public data is a fantastic just general strategy. I've mentioned the target product profile in the past. This is a sort of example simplified TPP from our CDK7 program. And we developed and continue to develop automated approaches to rationally generate these target product profiles. They're clearly different in different settings. Some targets, you need blood-brain barrier penetration for, and this leads to a sort of different physicochemical mask or sort of access strategy in the compound design. Sometimes you want to exclude a compound from the CNS, the brain, for side effects. So every target's sort of slightly different. There's a different mask or TPP possible for different therapeutic modalities. So injected peptides and biologics drugs are very different in terms of their TPPs to oral small molecule drugs and almost exclude and experience is that you can see many of the design goals are thresholds. So it's certainly not the case that a more potent compound is better than something just that achieves the exposure and potency required to modulate the disease process. So it's often one of the tricks in drug discovery is knowing when to stop against a particular parameter. So we bring all of these together into a multi-parameter optimization framework. This MPO acronym is very widely used in the space. And then we use these merit plots to really track and feedback on the sort of telemetry, the progress of every project. What we typically find in the early stages, where you're starting with a hit or someone else's lead, a sort of competitor disclosed lead, is that the merit scores are pretty poor. And then we look to rapidly sort of improve these towards our target function in this so-called exploration phase. And then we enter a slightly different, different phase, the exploitation phase, where we're really trying to tune the optimal combination of these parameters. And sometimes it's a little bit like whack-a-mole, where you have a couple of sort of required endpoints, numbers you're trying to hit that seem to be a little bit in opposition. And this is where the active learning and some of the deeper sort of SAR analysis methods that we've developed fit in. Another sort of general feature of ex-scientia sort of philosophy and also our platform is that we've tried to be, as broadly target applicable as possible, and we haven't nailed our technology to a particular subtype of AI algorithm. So we're very Catholic on the number or the type of AI algorithms we use. So Gaussian processes, Bayesian processes for decision making and so forth, models, random forests. We do a lot of physics-based molecular dynamics simulation, multitask learning, transformer models, and the sort of the real sort of workhorse of the majority of AI sort of deep learning methods. We're also very flexible with the type of data that we can use. Literature, SAR. We've got extensive experience in building and analyzing patent data. Again, one of the projects I was lucky enough to lead when I was at the EBI was the establishment of the world's largest online open patent-derived database that's built overnight automatically. And again, the nice sort of interplay for me there was that this was a sort of commercial product that the developers of donated to the EBI to put into the public domain. So again, a nice sort of interface between pre-competitive and competitive activities, which really enabled the work. Other examples of the data types we can process: high content screening data, so phenotypics. We do quite a lot of fragment screening, and of course all this is modulated by the prior knowledge and then the specific project related knowledge that turn these sort of global models and expectations into more active, into more local project specific models for particular parameters. This all comes together in a well engineered sort of model platform that we call Flux. We'll start disclosing some of that over the next 6 to 12 months. And then this leads to the generation of novel molecules. We apply learning, active learning approaches on top of this, and then experiment, synthesize, and then feedback into this generative AI space. So I don't know if you can see my cursor, but really, the looping around here till we get a compound that satisfies our target product profile criteria. So now I'm normally a little bit short of breath at this stage, so I'll show you a video of the sort of, capturing figuratively the sort of optimization that goes on, exploring a binding site, looking at all of these parameters in parallel to come up with a sort of a most optimal design given the constraints that you have around both the data and also the target product profile. And at the end, you can see this merit score moving up and down, and here we have figuratively a drug candidate highly complementary to the target, but also that's got good drug-like properties, lower toxicity risk, lower off-target interaction risk, and so forth. I mentioned physics approaches. We've put together a molecular dynamics toolkit, which is actively applied to discovery projects, both on the basis of say, docked structures, but also on the back of some of our in-house crystallography structural biology sort of studies, experiments. We do a lot of pose prediction. So one of the issues with docking is often discriminating real poses from decoy poses. We do some metadynamic simulations to address these. Potency predictions, both the relative changes, so whether an edit on a particular compound structure in a generative step increases or decreases potency, but also an estimation of the absolute binding energy prediction as well. We have in these automated, scalable physics-based methods. And the other issue is with, or the other factor that's important to us is on the back of lower cost computing and the scalability of computing and some of the systems that exist there. We're exploring different sampling methods, again, to address some of the drawbacks of earlier stage MD simulation approaches. Here we show the binding site moving, the shape and the properties and the pharmacophore features. And again, one of the lovely advances on the back of AlphaFold for the community is that now people are starting to explore binding site flexibility, comparisons between proteins and so forth. And also there's a number of companies using AI to develop approaches to find these cryptic pockets. Occasionally proteins have got a hidden target that isn't revealed until you've got a ligand. But they've often got really nice properties for selectivity or drug properties. So a nice example there would be a historical one of HIV reverse transcriptase inhibitors, where the NNRTIs, the non-nucleoside drugs, bind to this pocket that doesn't exist before the ligand's there. And again, I think the prospect and the excitement of data mining these very large data sets to find proteins of that class that have these cryptic pockets is a very exciting one. Automation. So in a way, there's little point in designing a compound in a day if it then takes 3 months to synthesize and test it. So we focused recently, over the past two years in particular, on integrating learning into a real, reduced to practice experimental and AI loop. So we have tight generation of the AI design with make and test, so the synthesis and screening of the compounds. And also one of the other advantages here is that the speed and the quality and the reproducibility of the data in this approach, has also got large benefits. So reducing the time down of the time and the cost of discovery is I think one of the huge benefits of this combined AI and automation strategy. And we're very excited about this this platform that we put together. So the platform, we've got a new automation facility in the Oxford area, 4,500 square feet, and a sort of automation studio. It encodes and automates the laboratory workflows, and it does this in a very modular way, in the same way that our AI platform is designed to be modular and scalable. What the automation facility does: chemical synthesis, compound management and logistics, and then the biological testing, which then feed into our AI algorithm for the next iteration in the design cycle. So this really does and increasing quality. And of course, not all assays can be automated. assay or in an animal model clearly has to be done outside of this loop. But again, the data can be fed directly back in into this design, make, test loop as you sort of move further towards the clinic and start to do preclinical toxicity studies and so forth as part of compound progression. So we have, it's a lovely sort of facility to visit. My mind is often blown, and I just stare, looking at these little robots running around, normally under their own sort of free will and volition, doing tasks for us. But if we've got very close physical coupling of the biochemical screening, and also on the automatically controlled way throughout the required part. Continuing investment for us. One of the other benefits and one of the emergent benefits of this sort of automation approach is that we can start to align the synthesis of the compounds with the capabilities of the automated chemistry platform. Not all chemistry steps, reactions can be done in an automated way. And now we score explicitly the synthesizability and the routes of the compounds prior to their selection for design. One of the other benefits of the way we've configured our platform is, I think, a sort of earlier worldview of scalable synthesis was really a sort of parallel synthesis approach where you have a common framework and then relatively little steps, little substituent variability in one sort of iteration. So common starting material, similar reaction conditions, and then minor exploration of SAR space. Our platform allows very much a sort of bespoke synthesis where we can do things like scaffold exploration, so changing some of the starting material, some of the cores, using different or being freer to use a different number of reaction routes and reaction types in order to maximally learn, going back to this sort of active learning information content approach for the SAR optimization, structure activity optimization of the platform. And again, a lot of the parallel systems were often single or very small number of steps. The ability to sort of iterate around allows us to have multiple reaction steps coming up with more sophisticated designs for the target compounds in this optimization cycle. The talked already about some of the sort of robotic connectivity, but I think there's a stunning video online, a collaboration between OpenAI, the sort of ChatGPT guys, and a robotics manufacturer where there's a stunning example now of some of the benefits of embedding AI actually in the hardware of the robots. So this video is it's worth activity from this sort of AI system that we've put together. A couple of gratuitous videos. I could watch this all day. There we are. So precision medicine, the sort of step to the patient. So I've talked a little bit about target ID, not much science depths under there. I've given a sort of a slightly deeper dive into the automation of the design and automation components and how that fits together. And then, how does this couple with precision medicine? How are the targets and the clinical trials we do sort of modulated or have higher probability of success? So our primary approach is to interrogate drug action to measure the effects of a compound in complex primary tissues. So in the context of tumors, this could be the cancer itself and the sort of stroma, the tumor microenvironment around it. We've built a best-in-class automated microscopy and deep learning system, again, an AI approach for single-cell analysis, a segmentation, and then measurement of various sort of cell parameters that allow us to uniquely profile the activity of compounds, or the activity of compounds in cells. It's both cell lines, but more importantly against a biobank of patient samples. A nice example of this, a specific example, drilling down to a specific patient, an 82-year-old cancer patient who'd become intolerant to chemotherapy. Primary patient tumor was obtained as part of a biopsy, and then 100 clinically usable drugs were applied to that sample, and then automated microscopy, single-cell image analysis happened, and then measuring the individual drug response on the back of each of those drugs in the context of the patient's real tumor. And this led to, for this patient, complete remission, 2-year survival, at a fraction of the cost of some modern therapies like CAR-T technologies. So this was put together and published as part of a trial where there was a significantly improved PFS for a cohort of patients on the basis of this platform, where the data was used to modify the prescribing data for the patients. So 54% of the patients had clinical benefit of greater than 1.3-fold enhanced progression-free survival compared to their prior therapy. So this data clearly, when taken in aggregate, allows us to generalize and stop and identify patterns across, say, genetic sequencing differences, copy number variations, all of this sort of stuff. And now our platform is really designed and configured to better understand differential or patient-specific response. We've got a panel of internal cancer agents, and of course, including our own development candidates and potential development candidates that we try against a biobank of patient samples, measure the data, and then try to correlate response with chemical structure and allow us to identify best-in-class patient activity, often where you have multiple drug classes working together. So this, I think, is a nice approach of getting AI methods and data science methods to really align therapies against patients, but also identify the general features that allow us to feed into target product profiles and even identify novel targets on the basis of patient response. So with that, I'll stop. Very happy to take questions. One thing I meant to say at the start is that Exscientia are an academic spin-out company, originally from the University of Dundee, and then moved the headquarters to Oxford, and now is primarily a UK-centered biotech. Thank you very much.
Thank you very much, John. That was great. I really like your examples. Really impressive video. The videos of the robots are really cool. So we have several questions from the audience, and I encourage people to submit their questions by clicking the Q&A button. But we'll start with Nina's question, who's asking if you could expand on the drug selection that went against central dogma? I think it was at the beginning of the presentation, you had some examples there.
In the field or the SAR expectations for these particular targets. I believe from memory, that example was to do with some of the physicochemical properties of the compounds that once we'd made this observation and confirmed it experimentally allowed us to optimize in a more direct way. But yeah, that's from memory. I can't remember what graph that example actually refers to.
Yes, thank you.
Is the dynamics of the target taking into account during docking? Another question from Nina. So yes, in a way. So maybe not a sort of very satisfactory answer. So we can run MD simulation docking studies, but I think the direction of the field is to do these sort of ligand protein co-folding. So we've seen some great examples, literally this week, last week now, maybe in Science, some of David Baker's work that showed that, this consideration of some of the induced fit into the docking process was sort of quite transformative, really. And, with a little bit of envy, I saw this week that an AI biotech with David Baker and presumably his technology on the board have just started off with a $1 billion sort of pile of capital and so forth. So really shows some of the belief in the community of the power of those sort of approaches. But again, it's never really one thing. It's the integration. It's the reduction to practice that is key. How do you make the compound selected? Is it a plate format? And how do you handle various reaction conditions in this format? So that is way beyond my competence level and so forth. Yeah, very happy to get an answer on that back. But as far as I know, so certainly the compounds are plated, but the reactions are done, one shot at a time, and we can configure different conditions and flow cell connections and so forth for that. But I'm filling in gaps a little bit like a hallucinating LLM there, to be perfectly honest. But very happy to come back with a sort of specific response there if you leave contact details somewhere. What advice can you give to a lab-based scientist, a chemist, to move away from the lab and get into using AI and data science for drug discovery? I think, my vision of the future is that there's going to be far less of a differentiation between sort of lab-based science and AI methods in the future. The smart people, well, the smarter and the most productive people will know some of the experimental practicalities and restrictions and be able to combine these with the questions they ask of the AI system. And my answer to the previous question was a lovely example, really, of how I'd benefit from deeper knowledge on the experimental side. It's clear that AI is transforming many business sectors and so forth. But the best advice I'd give to someone would be, depending on level of computational sort of competence and experience, start to use Unix, get to use like proper sort of computing frameworks and so forth. Python as a sort of programming language is a great place to start. You can use it for essentially as a sort of advanced, sophisticated sort of version of sort of Excel. But once you understand the basics and just get to the sort of hang of sort of algorithm structures and so forth, you can start to do a lot more. So I think Python notebooks, look to investigating data analytics approaches and so forth. I'd caution against diving into specific learning of various sort of AI techniques. A lot of our AI, the pace of innovation in the AI field is very rapid. These large language models, didn't clearly come from nowhere, but they've been very sort of, there's been a huge adoption and to some extent less focus on some of the other deep neural network methods and so forth. So trying to maintain it's almost like a Sisyphean task of trying to maintain cutting edge AI knowledge, because it's constantly changing. But I think the key thing is really to gain skills in how the various AI methods and algorithms and approaches can help this sort of chemistry or drug discovery problem. That's where I think that's where the sort of transitional potential becomes. I very much come from a sort of data background, and then started to really invest in my career in how AI machine learning could make best use of that data. So everyone's got different sort of paths to a career.
Thank you for that. I think we have some more questions. Yes. Someone said they love the example of the treatment of the 80-year-old patient resistant to chemo. How far away do you think we are from this kind of personalized medicine being available en masse?
I think it's a great question. I think fundamentally it's the embedding of this in the hospital systems is going to be the enabling factor. Again, time is of the essence. A lot of these very aggressive cancers have got short timelines, and some of the work that I've seen, some of the earlier work where you needed to generate organoids from a patient sample and so forth, unfortunately the results almost came too late to sort of assist the patient. So again, I think automation embedding in the healthcare systems is going to be the real answer there. Some hospitals are clearly doing that. My wife is a researcher here at MD Anderson in Houston, explaining my sort of physical presence here, and they're investing very heavily in that sort of omics integrated precision prescribing sort of approach.
Thank you for that. So what do you think? So you've talked a lot about what areas it's impacted, quite like AI, the discovery process. What do you think are the areas in the future that's going to be most impacted in the pipeline? If we imagine that slide where you have a pipeline, and the time you'll say everything, but if you had to pick one area, do you think that's the disruptive one? What would you say?
So yeah, that's a real tease of a question. So the industry clearly still has an issue with target selection and identification. That's a very major sort of problem. The trouble is that innovations there are quite a long way from the drugs, and they really just enable the specific drug discovery against those targets. And also in a business setting, it's pretty difficult to keep those sort of target choices private before others just jump in and sort of start working on them in parallel. So target identification's an absolutely wonderful area. I think it's clear. One of the companies I think is doing great work, again, Oxford-based, Genomics PLC, who look at sort of polygenic risks, so multiple genetic variations as being, or multiple causes behind a sort of a disease, and I think that's really exciting. People are probably most familiar with these sort of monogenic diseases. The sort of Philadelphia chromosome aberration in BCR-ABL that is treated by imatinib. That's a very discrete thing, but often chronic disease in large populations is caused by lots of factors all at once, and probably things like obesity are a sort of great example of that. So I think this, so target discovery I think is a great thing. Clearly, my and our near-term focus is laser-like around the design and synthesis and automation sort of pipeline. But I think there's also great potential in AI applied in the clinical space as well. So more efficient clinical trial design. I think now that biobanks, I do some collaboration with some researchers at the University of Liverpool on sort of real world evidence against sort of NHS scale records. And again, for me, this is just such a latent opportunity. And the more you try and link this sort of real world evidence to genetic and omic type things, UK Biobank again is a great thing. There's follow-on projects for that. So I think we're entering a really fantastic future. But again, one of the other dichotomies I think for the field is just thinking about the answer I just rambled through, is this difference between treating an individual patient and a population. And we know from many parts of biology there's emergent population effects, and fundamentally we want to treat a population, and treating that population as a series of heterogeneous individuals. So this multi-scale modeling and some of the impact on healthcare overall, I think, is going to be fabulous. So sorry for ignoring the question. If I had to pick one, I'd pick all three areas.
That's great. Thank you, John. That was very good. So I'm afraid we don't have time for any more questions, so I'll wrap up. So if I could share, I'll share my screen quickly just for some final notes. So thank you again, John, for that talk. That was great, and I'm sure the audience really enjoyed it. Just some last notes, as I said. A reminder to join SCI, and there's a lot of benefits to the membership, and we have a lot of events that might be interesting. If you found this one in particular interesting, discounted rates to conferences, for example, are of membership benefits. Future SCI talks, so similar to this one, we have a bit of a break until September, but then we have a trilogy of talks on dementia. So we have James Rowe from Cambridge University, Susan Corlas from AI UK, and Craig Ritchie. So I very much encourage people to sign up to these, which are going to happen in September. But with that, all that's left for me to say is thank you, John, for such a great talk, and thank you to the audience for that. So I hope you have a great rest of your day, and thank you again.
Thank you. Goodbye, everybody.
Bye-bye.